So good afternoon to everybody and welcome to today's webinar by Dr. Meera Venkatesh. Before we go to the webinar per se, I want to, since we have a large number of colleagues, friends, students and faculty gathered both on WebEx and YouTube, I just want to give a few announcements. One is we are on 20th, we will be having a lecture by Dr. Arun Srivastava, Secretary, Atomic Energy Commission and Department of Atomic Energy, about intellectual property rights. I am sure that will be a lecture of great value to students and faculty about taking forward their innovations towards exploitation. So that is on 14th, that is on 20th. And on 21st, we will be inaugurating an advanced course that will be given by Professor J.B. Joshi, Emeritus Professor HBNI and former Director of ICT, Mumbai, on design of multiphase extractors and contactors. And this is based on registration. Information about these programs are available on the Anuvidya website. And on 5th February, we will be starting a course on nuclear science and technology that will be of very broad interest to a large number of students across. So anyone who wants to join, they're welcome to access our Anuvidya website and register. That is being coordinated by Professor B.S. Tomar. So that will start from 5th of February. So with these announcements, I get back to today's program. My colleague, Professor P.D. Nayak, will formally introduce Dr. Meera Venkatesh. But before that, I must add that it's a great pleasure for me personally also to meet Dr. Meera over the web for this program. I just recall, I can see her smiling, I recall meeting her more than 4 decades ago when we were both young scientists around 1977 in the RLG of BARC, and both of us have come a long distance. After a very illustrious career in BARC, Dr. Meera also went to IAEA and made a mark in the international arena as well, and she's back in India now, and it's a great pleasure to have her. And I think it's a beginning, and we may be troubling her again to give maybe a more detailed course to many of our students, and I'm sure she will be happy to do that. So now I request, without taking much more of your time, I request Professor P.D. Nayak to formally introduce Dr. Meera.
Good evening to everyone and welcome to HBNI webinar. First of all, I'm thankful to Dr. Meera Venkatesh, our former colleague from BARC, for accepting our invitations to deliver a talk on multifaceted radiopharmaceuticals, the vital components of nuclear medicines for molecular targeting. In fact, most of the BRC scientists and engineers don't need the introduction of Dr. Meera Venkatesh. For others, Dr. Meera Venkatesh graduated from 20th batch of BRC Training School and carried out research at Baba Atomic Research Center in various areas related to peaceful applications of nuclear technologies, such as production of radioisotopes, development of radio labeling strategies, development of radiopharmaceuticals, and evaluation of radio-labeled products for their usability in hospitals. Between 92 and 94, she was a postdoctoral fellow at University of Missouri, Columbia, USA. And at that place, she worked in the area of novel molecules for use as the radiopharmaceuticals. Again, in 1999, she also served as a visiting professor at the same university. At BARC, she was heading the group of radioisotope and radiopharmaceutical divisions and radiopharmaceutical programs of BARC. At the same time, she concurrently was senior general manager at Board of Radiation and Isotope Technology, popularly known as BRIT. In fact, she has contributed immensely in radiation and radioisotope related technologies, and some of this has resulted into new products which have been used by in nuclear medicines in many hospitals in India. She received many awards in that excellence from Indian Nuclear Society and Department of Atomic Energy. After serving at BARC at various capacity, head of Radiopharmaceutical and Isotope divisions, from 2011 to 19, she moved to IAEA, as Professor Vasudeva just now mentioned about it. There she worked as a director of the Division of Physical and Chemical Sciences, and she was overseeing activities in multiple disciplines related to nuclear science, nuclear physics, accelerator applications, fusion research, nuclear data, radioisotopes, radiation technologies, and isotope hydrology. And our mission was to spread the beneficial applications of nuclear science and technology to global mass. In related to our basic research and other scientific contributions, she has authored over 200 publications in the international journals, book chapters, and she served as the editor of 2 international journals. She has guided 17 PhD students in the area of her expertise, and she is very passionate about promoting peaceful applications of nuclear technologies. She was a key member in the organization of the Scientific Forum on Industrial Applications of Nuclear Technology in 2015 and the Ministerial Conference on Nuclear Science and Technology Applications in 2018 at IAEA. Without taking much of your time, I request Dr. Meera Venkatesh to deliver her talk. Dr. Meera Venkatesh, please.
Thank you very much, Dr. Vasudeva and Dr. Naik, for those kind words. So I will not spend any more time on any of those formalities because I know also that the time is very less. Maybe I will share my presentation. Are you able to see the presentation?
2 minutes, madam.
1 or 2 seconds. Just hold on. Yes.
Yes.
Okay. First of all, good afternoon to everybody and a happy new year. And thanks to the HBNI Dr. Rao for the invitation to talk about the topic which I have worked upon for nearly 4 decades and it is very close to my heart. So that also means that I should apply my reins not to talk too much about different things and not to explain. So one talk now I thought we can be structured like this. It's a brief introduction for those who are really not aware of radioisotopes and pharmaceuticals, and then focus on radiopharmaceuticals to look at how they have evolved and what are the needs and strategies now which are being looked at, and how, most importantly, how so many specialties come together to launch a new radiopharmaceutical and how it is relevant to many people, not just people who work in that as radiopharmaceuticals. So this is just to have a placeholder. Sorry. So many people, all of you perhaps from the nuclear family, maybe you know that it is the first half of the last century, first 50 years, has been a golden era as far as radioisotopes and their applications are concerned. Here I have shown pictures of so many stalwarts who were instrumental in bringing out what is the radiation, how radionuclides can be used, and they're all Nobel Prize winning discoveries and innovations, which have led to the current state of so many applications being there. So, when we talk of nuclear radiation, we know that it could be it has to be coming from atomic nucleus, radioactive isotopes giving out radiations. And these radiations could be particulates like alpha or beta, or non-particulate photons like gamma radiation. Now, when we come to the radiations, one common thing is they are all having energy and they are transient and not permanent, with the concept of half-life being introduced. So all these nuclear techniques, they are all related either to the isotopic measurements or they are based on the radiation-induced processes. So again, I'm sorry for people who are experts in this field that I'm going into the basic things, because when I spoke to Dr. Rao, I understood that people from different areas which come under HBNI could also be interested in listening to me. So just to set the background, apologies if it is something very trivial. So why and how are radiation useful? As I said, the primary thing is they are energetic, they are powerful, they can cause changes when they come across a new material, and they also get attenuated in that process. So because they are easy to detect, they can be like spice in the information. They can act as tracers, and they can kill germs, cancers, bacteria, viruses, etc. So they have this damaging ability to detect. You can also the materials, whether it is non-inanimate, or it could be also the human tissues, and they can break the bonds, they can make the bonds, so many characteristics just because they are radiation. They can also provide information, like here I have shown whether it is in the border security or whether it is in a museum, they can provide information by passing through the material. And of course, there are certain unique applications like being source of energy. But we are not going to talk of all that. This collage was given just to give a bird's eye view that there are so many numerous applications that are so different from each other. And when we talk of radiation-based technologies, they can, and as I said, they can be used in different areas: healthcare, agriculture, industries, environment, water resources, and whether it's art, artifacts, of course, research and development, and miscellaneous applications. As I said, this collage is to inspire you, whoever is from the younger people who are planning their careers, to look at the various applications and to be inspired and work in one of them or many of them. Coming to the healthcare applications, as I mentioned, radioisotopes can be used to give information as disease cases, which will be useful in diagnosis. They can be used for therapy because they are really tedious to use. They can be used also to look at how a patient is doing after treatment. Again, diagnostic imaging, sterilization of products, drug research. Drug research has hugely benefited from use of diagnostic imaging techniques and certain high performance materials which can be used in healthcare. But among these, the ones on diagnostic therapy, monitoring, and drug research use the nuclear medicine techniques and radiopharmaceuticals. So what are radiopharmaceuticals? They are the specially labeled molecules which are used inside the body in nuclear medicine. It could be injected, it could be taken orally, whatever. But the unique feature about nuclear medicine and radiopharmaceuticals is known as molecular imaging. It gives you the functional aspect, functional information, as well as anatomical information. And this is happening because whatever is being made is done through the molecular mechanism most of the time. When we go to the evolution, we see how things have evolved. But right now, it is primarily molecular interactions. And when we talk of radiopharmaceuticals itself, they have two components. One is the radionuclide, and the other one is the molecule which is carrying the radionuclide. So here I have put two green things. This is something like a clamp holding on to the nuclide. This right now I'll just introduce them as chelators or chelators, just to hold the radionuclide to the biomarker. That will be useful in understanding what we talk later. So when we look at radio tracers for diagnosis, you have it is administered, radiopharmaceuticals administered as image. When we talk of image, it has to be photons or gamma rays. And this could be coming out from radionuclides which emit gamma rays, or it could be annihilation photons from positronic. This is why you have so many isotopes are either gamma.
Dr. Meera, sorry to interrupt you for a moment.
Yeah.
I think you need to keep your mobile away. Maybe if you are having it nearby, because I think there is some disturbance coming because of that.
Is
Second,
it okay now?
it's okay, better now. And can you hold your mics little closer, and then voice will be even more clear.
Okay.
Otherwise, it's fine.
Okay. I'll just.
Sorry for interruption.
No. It's okay. Yeah.
I can hear the disturbance when your mic moves.
How about now?
That's very nice.
Okay. I'm sorry.
Okay.
I should have checked it earlier with you. Okay. So when we talk of diagnostic nuclear medicine, we are using the radiations which are imageable, like photons from gamma rays or annihilation photons. I have just mentioned a few isotopes here. And primarily, such diagnostic imaging is used to look at the anatomy or function of organs, tumors, shape, position, etc. And cardiology and oncology are the most used, the two specialities which use nuclear medicine in a big way. Because as you can see, the data from WHO, lot of deaths occur because of cardiovascular diseases, and the next one is cancers. So, but other specialties like neurology or infections and liver, kidney studies, so many others, plus drug research, use nuclear medicine in a big way. Here I have just shown a couple of scans of thyroid. Even a non-specialist can look at a normal thyroid and see something is wrong with it. So the beauty is that it speaks, the images speak a lot of information compared to what one can find in different other kinds of investigational diagnostic tools. So these are extensively used in cardiology, oncology, and many other specialties also. And here, I have, as I mentioned, it can be used to find out the shape, size, abnormalities in organs by what is known as static imaging, just to look at the images of the organs, or what is known as dynamic imaging, where one can look at the functioning, like in the case of heart, liver, kidneys, etc. The functions can be measured. So the diagnostic imaging gives a lot of information for finding out what is wrong in the patient to the doctors. Here I have just shown a heart scan, the cardiac perfusion scan, what is known as how the blood flows. It gives a lot of information to the doctor to see what part of the heart is working, how much part is not working, whether there is a fault in the valve, etc. So this is just to give a feel how this works. This is another set of pictures which clearly says a normal person, a phosphonate, when it is labeled with when it is labeled with a nucleate like technetium, how a normal scan looks and how a scan in a cancer patient with metastasis or spread into the bone looks like. The positron emission tomography, I mentioned to you that the annihilation photons can also be imaged. In fact, the annihilation photons being two of them, they give much highly resolution images, which is very useful in looking at the cancer lesions, etc. And it is sought about by the doctors very much. So here you can see those images. And in therapy, when we go to the therapy part of it, it is the administration again of radiopharmaceutical for therapeutic effect, which means the radiation has to be high linear energy transferring radiations, which are usually the particulate emissions like beta emitters or alpha emitters or Auger or conversion electron emitting radionuclides. There are a lot of nuclides. I am not listing them here right now. We'll look at them later. But the point here is that when a radiopharmaceutical goes to the tumor, after it reaches, you see that different particulates have different tissue ranges, and depending on how much of volume has to be irradiated, one has the choice of using the right kind of radionuclide to have the therapeutic effect. It's a very well established, in fact, therapy was established even before the imaging things became very common. Radioiodine, as soon as it was available, it was used in treatment of thyroid cancer, etc. and primarily it is therapy of cancers and could be pain palliation in cancer patients. The non-cancerous ailments like hyperthyroidism and joint disorders can also be treated with radiopharmaceuticals. That's being done. So nuclear medicine has grown in a spectacular way through the 20th century. Here again, I have put a collage of a lot of people who were very important in bringing out, having discovered and innovated many things like scanners, gamma cameras, finding out the new isotopes. Seaborg will be a common name to many of us in this group of radiochemists. And also the invention of what is known as a generator. And apart from the radionuclide area itself, the innovations in the biology area, like monoclonal antibodies, tumor-specific peptides, all these ushered a lot of acceleration into the nuclear medicine in its growth. And I will mention just a little bit about technetium. Many of you may already know about it, but a generator or a cow is a great way to avail an isotope. Like we know that isotopes can be produced in reactors or accelerators, but having a generator makes it so much more convenient in a hospital to make the radiopharmaceuticals, to tag them and then use them in the patients. But the unique thing about technetium is its characters, that it can be imaged in a beautiful way using single. I will come to it later maybe, but because of its unique features, it has been named as a workhorse in diagnostic nuclear medicine, with almost 40 million studies being carried out per year globally. And fluorine-18, which is the PET image or the positron emission tomography image, is soon catching up. In fact, some of my medical fraternity friends tell me that it is even it's gone. It's in their hospital, the use of FDG PET is more than what technetium is being used. But I'm using figures which are from from the publications. But definitely PET imaging using fluorine is also a huge numbers. So this is Dr. Wagner, who is no more, but then he was known as the father of nuclear medicine in the US. And he named FDG as the molecule of the millennium, last millennium. And because of the advances in the practice of radiopharmacy and transportation of radioisotopes, also these have helped in the growth of nuclear medicine. So, as I said, in the past decades, a lot of growth has been there due to the innovations. One is imaging technologies. We saw that there are, even as people who are working with radiation will know that the kind of detectors, the processing, computation have grown so much that starting with the gamma camera, which can only image a gamma, we had single photon emission computed tomography, which is known as SPECT, which gives much higher resolution than just a gamma camera. And positron emission tomography is even better resolution. So that is on one hand about the technology of imaging. Then it's added to that is the fusion of images from other modalities like computed tomography or MRI. So this kind of fusion helps in combining the information which is available only on anatomical basis, which are very good with CT, and adding the molecular angle to it through the PET. So this gives a very, very useful and very valuable information to the treating doctors, even if they have to go for, let's say, radiation treatment, external beam radiation. These kind of images are now being added on to the treatment planning so that they can irradiate the right area. It need not be always treated with therapeutic radiopharmaceuticals. They could be treating with external radiation or other modalities. And one is the imaging technology. The other is advances and growth in the radiopharm molecular biology, because this has really fed into the growth of radiopharmaceutical, because one can know about how the cancer grows, what are the markers, and how they can be targeted. So there are a lot of molecules which have come up in the last 40, 50 years, which gave an insight into how to reach the tumor, how to target the tumor. So these innovations have helped a lot in the growth of radiopharmaceuticals. On the one hand, the biology part and isotopes also. isotope production, availability, and the radio labeling techniques have also aided in the growth of nuclear medicine practice through the right radiopharmaceuticals. So if we look at the radionuclides used in the past, phosphorus and iodine were used long ago. And over these years, as I said, a lot of growth has been there. One of the big paradigm shift was when technetium-99m became available. The next one was when fluorine-18 became available. Although it was known long ago, before this big surge in its use, it became available for very, in a wide way in clinical practice because of the availability of medical cyclotrons in a big way. And many nucleides with great promise, whether it is for imaging or for therapy, they are now gaining prominence and are being used in a big way increasingly. And focus on therapy is growing very steeply, and use of alpha emitters is seen as promising. Once upon a time, alpha emitters, everybody would have thought, oh, it is very huge, it may not reach the lesion, it will destroy the tissues on the way. But now the results and the way the work now which have been reported are extremely impressive, and people are working on alpha emitters for therapy now. Not only working, they are using it for therapy. When we look at therapy, its ambit is also growing because of targeting molecules, and a huge number of cancers are now possible to be very specifically targeted. Non-Hodgkin's lymphoma or neuroendocrine tumors are being very well treated using radiopharmaceuticals. And as I mentioned, a lot of isotopes are also being explored with impressive success. Lutetium, yttrium have been there for quite some time, and they have established their own niche place, especially lutetium. Actinium, the alpha emitter, is a rising star now, and many more are there in that list. And the idea or the focus now is on personalized therapy. What we mean is the therapy is planned based on the diagnostic imaging. They are closely linked, and the word theragnostics has been coined, although iodine-131 therapy was always preceded by an imaging using the same, and you could call that is the beginning of theragnostic idea. But here now, the idea is to use the same biomolecule, the same targeting molecule, to be labeled with either a pair of nucleides, either of the same element or similar elements, so that a diagnosis can be followed by therapy in order to know proper localization, accurate dosimetry, and judging the therapeutic efficiency. So this is the way the therapy is moving, and people are now looking for personalized therapy so that it's much more efficacious. So in the case of radionuclides, again, this is not an exhaustive list. I have just put some of the commonly used or something which it's not always commonly used, but I have highlighted the ones which are regularly used and which are approved by drug authorities, etc. So you can see that the number of isotopes which can be used is huge, and some of them have very good properties in terms of their behavior or in terms of their radiation properties, etc. But not all of them find the way into the clinics because of many other reasons, which I won't dwell now. But the people who are the young ones who are looking at a career in radiopharmaceutical or something around it, they have these challenges or these opportunities to look at what else is possible, how else we can aid in nuclear medicine to bring even better radiopharmaceuticals. So I had mentioned about theragnostic radionuclides. We can also call them as surrogate pairs, where one helps in imaging to calculate the dose and the localization, the other helps in the therapy. So here I have again enumerated the beauty of technetium, because it has really ruled the diagnostic imaging scenario for a long time. And even now, I believe many of the hospitals still use technetium for the diagnostic imaging. So I had already mentioned about its excellent properties of half-life. It emits single photon, so you get a good tomographic image using SPECT. And a wide range of radiopharmaceuticals are available to image almost all the organs of the body, almost all the functions also. Maybe there are a couple of them where still further people are working, whether you can get a glucose analog to image heart, etc. It continues to be the most used diagnostic radionuclides. And the global shortage, which was experienced a little more than 10 years ago, led to innovative ways of looking at its production, use, and using alternate radiopharmaceuticals also. So even a crisis became an opportunity to look at other isotopes, other radiopharmaceuticals, other methods of producing. So I think that is also a way to look at radiopharmaceuticals. Okay, something is working, but what else can be done to make it even better? So here is the FDG. You can see the beautiful images obtained from positron emission tomography. So as I already mentioned, it's also used in a very large way. It could be called as the most used PET isotope, workhorse of PET. The therapy focus I have also mentioned. You can see here that there is just a small diagram to show Auger electrons. If you want Auger electrons to cause therapy, it has to be right inside the cell, which has to be killed, almost close to the DNA. So usually Auger electrons are labeled with nucleotides, etc. Alphas will have to be very close to the surface of the cell or very close to that. Betas can irradiate several cells and huge thicknesses of tissues, range in the millimeter. So here, now a wide range of therapeutic radionuclides are available, and molecules to target are also available. And the production, separation, and availability of radionuclides also gave rise to it's like a fillip to the use of radiopharmaceuticals for therapy. And many isotopes have even been revisited in the sense when once you have the proper molecules, maybe isotopes which were abandoned long back can now be thought of again to come into play. So I just thought I will give a quick perception of the efficacy of therapeutic radiopharmaceuticals through some images. Now this picture you already saw. When one phosphonate can be used for diagnosis, another phosphonate labeled with therapeutic radionuclide can be used for palliative therapy, which is what is done now, which is being followed in many places. Here is a picture of a prostate cancer patient where you can see that the prostate-specific antigen levels were very high and a lot of metastatic lesions were seen at one time, was treated using a peptide which binds to the prostate-specific membrane antigen labeled with an alpha particle. So with 3-time treatment, you can see all these dots have vanished, and one more treatment, even the prostate-specific antigen level has become very low, almost normal level, absolutely no sign of illness. So this is the kind of, it's very gratifying both for the patient and for the doctor if such results can be obtained. So that is one, that is why it is also inspiring to look at what else can be done to help the patients to come out of the disease or at least be palliated. So here, this is another patient with liver metastasis, huge one, treated with lutetium as the therapeutic nucleotide, therapeutic nuclide, and you can see that liver mets have gone. This is another one, prostate-specific membrane antigen as the targeting, the peptide which will bind to the membrane antigen of prostate cancer is the molecule, and labeled with gallium for a PET imaging, which you see here, all these red colored dots. These are the images before treatment, and after treatment with the therapeutic radionuclide with the same molecule, you see these patients are almost free of the disease. So this is the way the diagnostic or the following the therapy with diagnosis is going on now. So how does a radiopharmaceutical act? How do they work? So the mechanism is not single one. I have talked a lot about antibodies, peptides, etc. That's not the only way. There are multiple modes which have been used, particularly in the initial stages. The radionuclide as well as the molecule labeled play important roles. The physical properties and the chemical properties, charge, structure, lipophilicity, stereochemistry, all of them are important. And because of the complex nature of once it is radio labeled, the molecule is different. It's not just the radionuclide, it is the whole thing. And there are radiopharmaceuticals which are known as radionuclide essential, and there are radionuclide independent radiopharmaceuticals. So in the beginning, simple molecules like iodine as iodide were used, because iodide, as all of us know, it goes to the thyroid. It was used for imaging as well as treating thyroid disorders. Phosphorus, just as phosphate, was used in treating some of the disorders. Then later, there was also simple biochemicals. Here I have written genuinely labeled molecule, which means that this molecule, when it goes to kidneys or liver, wherever it was used, they already had an iodine atom, which was replaced with a radioactive iodine atom. These are called genuinely labeled molecules. They were used in the past, even when as Dr. Rao said, in 1970s, these were products which were supplied by BRC to many, many hospitals. And I have worked on them, and it is now looking like a very old history. Nobody perhaps uses them now. But these paved the way to new molecules that were tuned to suit the needs, like based on the hydrophilicity or ionic nature, lipophilicity, etc. So I'm not going to read all what I have written in my slides for saving the time, but I'm sure you can get the PDF version if anybody wants, or you can write to me if you need some more details. I mentioned about radionuclide essential. I have just given two examples here. This is isobutyl isonitrile complex of technetium, which is a +1 charge. This goes to the heart. This can help in imaging heart. And this one, if technetium is removed, there's no radiopharmacy. It's not like a biomolecule which will anyway go to the target. No, these are essential radiopharmaceuticals which will go only if the whole thing is like this. Also in the case of another complex which goes to the brain and things like that. So here, one can, if a person is looking at radiopharmaceuticals, there are radiopharmaceuticals which are very much dependent on what metal center or what radionuclide is there. Only then it will behave like a radiopharmaceutical. But there are also radiopharmaceuticals where I had shown you that there is a biomolecule which will go, and radionuclide is just tagged along. It is literally tagged to pull it along and going to the target. So to tag along, you need these kind of structures which are called chelates, and DTPA, which is a very common molecule, many of you know. This is one of the chelates. You can see like a claw. They can hold to the metal center like a claw. This works very well with the particularly the metallic radionuclides. With other maybe halogens and all, it won't work. But right now, when we talk of radionuclides and a variety, many of them are metallic ones. Of course, iodine, fluorine, you leave them, but other than that, most of them are metallic. So importance of chemical sequence. See, I mentioned about hipuran. So if you look, this was used in the past, but the chemists, the people who worked on developing radiopharmaceutical noticed that a certain kind of chemical moiety is required perhaps to go to the area where the other one was. So this one you can see the three molecules have some kind of commonality, and all of them are could be used for tubular filtration function of the kidneys. So this is one way of looking at radiopharmaceutical, what works and what can be made better, what should be retained and what should be improved. The charge, I mentioned already about the charge in the case of technetium, this isobutyl isonitrile hexakis compound. So this +1 charge helps to go to the heart, which is some of you may know that potassium plus as an ion has a role in the heart imaging, the functioning. So that property is used to look at heart through a positive +1 cationic complexes or molecules. So here for kidneys, they can be negative. Neutral complexes can cross the blood-brain barrier, lipophilic complexes through hepatobiliary. So these were the strategies, these were the way of thinking, and this is how things developed. And when it comes to the receptor-based binding, stereochemistry is extremely important because it is a three-dimensional binding to the receptor, and it is important that the complex or whatever is being used has to be of a certain stereochemical nature to give you the image or to go and retain in that organ. So for example, here, this is dimercaptosuccinic acid complex of technetium. Both are same molecules complexing, but the technetium here is in +3 state and +5 state in this. And they behave so differently. They look different, they behave differently. So it is important to pay attention to the stereochemistry when it comes to small complexes like this. When it comes to the binding portion, if it is going to be the radiometallic portion itself binding to the organ or the tissue, then stereochemistry plays a very important role. So I just thought I will, because cardiology, oncology, neurology are the ones which are primarily using nuclear medicine in a big way. Just a quick bird's eye view for looking at what is done. Heart agents, maybe perfusion. Just they want to know how the blood flows. The image which you saw earlier. So there are many components, many compounds being used. Technetium maybe is the most widely used, while thallium as a +1 is taken as the gold standard. But a metabolic function, if it has to be done, then one needs to look at metabolism of the heart, and fatty acid labeled with the isotope is used. So it is also important to know what we are actually aiming to look at. What is the aim of the doctor? So this is where question of understanding the biochemistry, talking to the biochemists, people who are already very well aware of that, helps a lot in developing new molecules. So heart and maybe brain have already been well addressed, or at least to brain still not, but heart is studied very well. But there could be other areas where if there is an imaging needed, understanding of the biochemistry helps a lot. So in the brain agents, I'm not going to again read out, but there are some agents labeled with technetium which go to the brain. But you see the mechanism is also quite tricky and smart sometimes. Like for example, this complex, it's an ester. After hydrolysis into the brain, it gets trapped, and that's why you get a beautiful image. But this worked only in the primates and above. So one has to be, when developing, I will come to that at a later part, but it's not as simple just to do some experiments one by one, but one has to keep the mind and open, and also think out of the box while developing new radiopharmaceuticals. So people have also looked at the functioning of the brain using different kind of molecules to, in the case of Parkinsonism, etc. And here I have just shown how they delineate. This picture, I wouldn't say that everybody can read, but it gives you the it gives the information how to look for abnormalities by using these kind of nuclear medicine imaging. And disorders of brain because of old age and seizures, schizophrenia, etc., are still matter of lot of studies. You can see how people are looking at the brain functioning through understanding the biochemistry. So this is just a picture which I took out from one of the publications, but you can see the thinking one step ahead, one step backward to see what is the molecule which goes into these kind of disorders, what can be giving you the information. So that is about imaging brain. And looking at other than simple organic molecules, then came the use of target-specific molecules. I have already spoken about this, antibodies, peptide, etc. But it is important to have molecules which will be molecular, which will interact in a biochemical way. So here, advances in the cancer biology, biotechnology, and molecular biology have helped a lot. Here I have just shown a cartoon of the various trapping mechanisms of a cell. You know, it can be so many cells itself is like a big world. There are many mechanisms, and one can look at whether you use an antibody or its fragment or hormones, drugs, peptides. So there are numerous possibilities to attack or to go and stay in the cells, to go and bind to a cell or go inside a cell also. So if you look at from another, those are kind of the strategies used for targeting. And if you look at from a different angle, initially it was simple biochemistry. Physical trapping, when it is not possible to target through some chemical or biochemical means, people have also used, people in the sense the radiopharmaceutical community, nuclear medicine community have used physical trapping as a mechanism. For example, for lungs or the joints, liver cancer treatment, reticuloendothelial system imaging. I have just mentioned here particulates. So this is just a physical trapping. If it cannot be done with a chemical thing, at times this helps, and this helps in a very good way. So these we have already seen about affinity binding using peptides and antibodies and other metabolic tracers. So that was about the molecule which goes and binds. Radionuclides, yes, the advent evolution has been due to the availability of radionuclides in a big way. Huge surge in medical nuclear cyclotrons, which have now been there, and their capabilities, because it is not, in the past 20-30 years, the number of cyclotrons we use for medical isotopes has really increased in an exponential way, and their capabilities are also, and then even now, there are a lot of cyclotrons which are being built for, particularly to give radioisotopes for nuclear medicine, but with special capabilities to give some exotic isotopes and isotopes which require a huge amount of capabilities, high energy and separation, etc. So here, this is from the cyclotron point of view, but we also have people who are making the targets, enriched target availability, the chemistry of isolation, automation of the products. You can see some of these pictures where the automation is so much that it cannot be done just by one chemist or somebody who is working in radiopharmacy. It has to be a huge team where there is somebody who is looking at the reaction, somebody tells what has to be added one after another, and how the things can be shielded, how it can be operated remotely, and then you get the product. So this is just to give an idea about the radionuclides have come a long way because of the support and input from several other faculties, automation, engineering, robotics, etc. So here I have just, it's not only that, it's also the chemistry, separation technologies. Separation technology is a huge science, as many of you know, and new technologies can be tried. It's very vital for purity. Can be, otherwise chromatographic column and some separation, many people, it has been used. But there are also technologies, like for example, here I have shown liquid membrane separation. This I had the fortune of working to separate yttrium from strontium, which was used in the back-end fuel cycle. It was back-end fuel cycle and radiopharmaceutical need not be together all the time. But if we can use the technology which was developed in some other place to look at whether it can be used to separate some isotopes in a high purity, it really pays. It really is very useful, and it's very satisfying also. So separation is a very important thing. It could be this, or in the cyclotron production, separation is also very important, how one removes the isotope from the target, and exploration of novel or alternate nuclear reactions. So here I have just given one example of lutetium, which is a very important isotope, and because of it can be produced in huge amounts because it has a huge absorption cross section, but it is only present in 2.6%, and this can be enriched. So one is enrichment of isotopes. The other one is looking at alternate technologies where you can get very high specific activity, because in many of these radiopharmaceuticals, specific activity is extremely important for going and giving, reaching the target and giving the required effect. So looking at alternate reactions is also a very important factor in bringing a radionuclide to be used in a feasible manner, in an economic manner, feasible manner. So this is just one example where people are now looking at this route because it gives lutetium, which is very high specific activity. Not only that, it avoids a metastable lutetium in this. I won't dwell more into that, but this is another example where germanium, gallium-68 is obtained from germanium as a from a what is known as a generator, but a direct production of gallium through a say cyclotron is also very attractive for a institute which has the cyclotron, because the generator could be very expensive. So this kind of other thinking has led to novel reactions, trying out different reactions to avail radionuclides. So the other aspect about radionuclide and the evolution is the radiolabeling strategies. This picture I have shown you several times. The chelating part, the linker, the biomolecule, which goes and sits on the receptor. Primarily, it is used in tumors. So here, the agents to secure this chelating moiety is also very important, because it is not that what works for gallium will work for yttrium or anything else, technetium, because each one of them, their size, what works for copper may not work for, gallium or indium. So people will have to look at the charge as well as the size, etc., when it comes to binding the radionuclide. So the one is the novel agents and also the automations to prepare these things. So the chemistry of the radionuclide is a very important factor in this. I have just tried to show some of these chelating agents which have been used in a big way. Here there is a biomolecule labeled with gallium, but you need such a huge thing to carry it to the biomolecule. These are some of these macrocyclics which are covered, where the space in between has a certain size, and that will determine whether a certain metallic nuclide will sit very comfortably and nicely there or will not be useful. So here, without wasting time, you can look at the complexity of the molecules. This is a peptide, but when it has to be labeled with a metal center like technetium, you need different kind of, so it also depends on the metal center, whether it is a soft metal or a hard metal, whether oxygen, nitrogen will work or sulfur will work, whatever. So this kind of organometallic chemistry helps a lot in developing the molecules which can bind to the radiometal center. So the biomolecules have had a great impact on the radiopharmaceutical. This I have already mentioned about antibodies, peptides, etc. But it's not just knowing what the biomolecule is. It is also the large-scale production. If something has to be used continuously, it is necessary to get it in huge amounts. So the monoclonal antibodies developed by Kohler and Milstein, which was Nobel Prize winning technology, had a huge effect. And it is even now, a lot of antibodies are used just without radio labeling for treatment of cancers. And among them, many have been also radio labeled and tried, out of which a couple of them are in the clinics as a regular product. So understanding the biology and expression of the cancer cells with receptors for peptides has helped a lot in developing radiopharmaceuticals. So here I have listed so many antibodies. Not all of them have become commercially available radiopharmaceutical. The two of these have become, and they are useful in many cancers. Although there are 45 FDA, I mean, I'm using the US FDA approved antibodies. So here you can see that when there are antibodies labeled with radio tracer, they can go to the antigens and bind to them and kill them. And it is primarily, these two both are used in B-cell tumors, so non-Hodgkin's lymphomas. This one, I just wanted to say that, okay, antibodies are one, but they are huge. So people have worked at least for 20-25 years to look at whether they can be cut, only the binding portion, small portion, engineered fragments. These are also huge areas of research. And I just wanted to give a pointer to this, because if there are people from the biotechnology, this area, who want to look at what else can be used, and the antibody fragments which are engineered to be used in radiopharmaceuticals. This is one area which is being followed, but it has not given spectacular results. But there are a lot of results which are very impressive. So this is one way of looking at molecules. Okay, there is a molecule, how best we can alter it. Even the receptors, when they bind to peptides, people look at how smallest the peptide can be so that it can go around in the body and reach the tumors. This is a list of peptides I have given here and the cancers in which they are treated. The somatostatin peptide analog, or the small part of it which has been radio-labeled and used, are octreotide and lanreotide. I'm not going to again read these, but huge number of peptides are also found to be very specifically linked to the tumors, and they can be used as radiopharmaceuticals. So potentially tumors have lot of places like fingers which can be bound with different kinds of molecules. One is people will have to find out what molecules, whether they will only go to the tumors, and whether we can attack them with the payload of radionuclides so that we can get a good amount of therapeutic effect, as well as label it with a diagnostic nuclide and see where and how big the tumor is. So when it comes to development of tumor, the needs are that it has to target with very good specificity, and oncology, neurology continues to be major areas for developing new things. Infection imaging, particularly for countries like India, it will be very useful. And the thing is, we want molecules which will quickly reach and bind to the target and not bind to non-target. It's very nice, good wish list, but it is quite challenging. And it has to, if it's a diagnostic thing, it should not stay anywhere else. It should just go out of the body and give a very good high resolution picture. So these are wish list, and targeting molecules, people are again looking at knowing the biology, biochemistry, and not only that, also manufacturing. So there are some of the radiopharmaceuticals are looking at something which worked long back, going back, and one of them, like copper 64, as just a copper +2, cupric chloride, sorry, has shown excellent result in treating cancer resolution. So it need not be always very complicated. One will have to keep on looking at multiple options and see whether something very simple can also work. So keeping the basic needs in mind and looking at various strategies helps. So the current trend is use of short-lived radionuclides. Long ago, people used to think that if it is only 1 hour, 2 hour, it is too short. But now, after having used fluorine with 110 minutes half-life and even shipping it elsewhere, people's mindset has changed, and isotopes which were discarded long back will find a place again sometimes in being used, because they may have very good chemical properties and physical properties, radio labeling properties. New nuclides, radionuclide generator systems are very attractive also, because they can help in getting radioisotopes in the clinic very easily. New biomolecules, as I said, personalized therapy using Diagnostic, and actinium-225 results have been very impressive and is pursued very avidly. If you look at the periodic table, lot of metallic nuclides are being used and being explored further and further so that they can be made into radiopharmaceuticals. So people who are very good in organic chemistry, organometallic chemistry, will find ways how to bind them to the biomolecules and have very good radiopharmaceuticals which are stable and which can also go to the targets. So here I'm going to put several tables. I'm not reading them. I'll just quickly pass. It's just to give you an idea. There are a lot of isotopes which have been explored, which are being used, and the whole ground is very huge and open for many people, many explorers to look at how best to use a different isotope, how best to produce them, how best to label them, how to test them, and try to use the best part of the radionuclide for developing radiopharmaceuticals. In this technetium, of course, I have highlighted, which is very, very widely used. Here, positron emitters, plenty of them, so many positron emitters. And I have also given the primary routes, because it also gives an idea about how complex it can be, what kind of target is needed, etc. The short-lived isotopes, of course, have very short-lived, 2 minutes and all. It's very difficult, but people are still attempting to use them when the cyclotron is next to the hospital. So generator system, as I said, are very useful. In the PET generators, some of them are being avidly pursued to find out how to make these generators available to the clinics. So when we talk of radiopharmaceutical bench to bedside, its processing of radionuclide production, separation, etc., choice of the biomolecule, radio labeling and its purification, evaluation. It's not just producing, because it has to be done, physical, biological, animal models, animal imaging, etc., and of course clinical tests. And wherever possible, if it can be made into a very quick formulation product, that's also very useful for its widespread use. And that is where multiple specialties come into play in launch of a new radiopharmacy. It's not always very clearly visible, but there is a huge amount of specialists who are there. Cyclotron, reactors, experts in target preparation, irradiation, separation, isotope production specialists, of course, separation chemistry, not only, not just the simple ones, but sometimes very unique way of separating helps. I have worked with some of the separation chemists from BARC. It was always a very interesting and enriching experience to learn how different molecules can just pick out what you want from a huge mixture and give you. So those kind of knowledge and experience helps. Automation, biologists, of course, microbiology, biochemistry, molecular biology, cancer biology, animal house veterinarians, clinicians, naturally at the end when things all go, and then finally, and the medical physicists and the physicists as such, who can do dosimetry calculations and give you the idea about what is how things are working. All these specialties come into play when we talk of radiopharmaceutical being developed and put it into clinical use. So there are attractive nuclides with challenges, like, for example, double neutron capture, reaction that need very high particles, whether electron beam accelerator can be used, alpha emitters, how to separate them from nuclear materials that they are already there, whether we can get isotopes from which is already said as waste. These have been explored already, but these are again opportunities because it's not become a common thing yet, but there is still a possibility. And production of parent nucleides for generator system, all these things are definitely opportunities for widening the roles of radiopharmaceutical and ambit. And I have just shown the decay schemes here. People who are working with these actinides will know this by heart perhaps. But then if we can separate actinium-225 or thorium-227 or actinium-227, it will be a great thing. It will be a big boost for our country to start targeted alpha therapy also. So these are challenges as well as opportunities for our scientists. And it's definitely a multidisciplinary activity, and it has to be with seamless working and collaborations and cooperation. I think in the current period, there is no seam between even the different faculties. Biology merges with physics so easily, physics with chemistry so easily. So they are all multidisciplinary activities where people will have to work from different expertise together and with collaboration, cooperation, deep involvement, and also with keeping the larger picture of, say, serving some kind of a bigger service, and that can be a very satisfying thing. So with whatever little opportunities I have got to work with doctors or other people, it has always been at the end some kind of a satisfaction. I think that is the most important than having the name as the first name in the paper, etc. That's okay. But then I think this bigger picture will have to be kept by the younger generation to look at what more can we do, how an automation person can help me to make radiopharmaceutical things like that. So with that, I hope that our younger scientists and experts can expand and work together and make a lot of progress and bring out lot of products for our our healthcare professionals, our patients, and be very satisfied also at the end. And I thank my teachers and mentors who paved my professional way. And of course, definitely I can never forget Bhaba, the training school experience, the working at BRC and BRIT, all my colleagues who helped me grow. And our IAEA work gave me an opportunity to peep at how the global scenario is. And definitely my family and friends and colleagues for the encouragement, and all of you for listening. Thank you very much.
Thank you, Meera, for a very exhaustive presentation and also for highlighting as frequently as you can that this is really a multidisciplinary activity. And it can be really quite a challenge for a group to put up, a group of starting from chemists to organic synthesis people, to separation chemists, to biologists, and to those who are familiar with the medical fraternity, clinical trials. I think it's a very long way. How in a department like DAE, I think this comes very naturally because all these faculties are around. But how do we otherwise people do collaborate across institutions? As you have been in IAEA, you will know what is the strategy adopted in other countries. Suppose some student wants to look at a problem and he has only one specialization within himself. So how does he go about doing it?
Yeah, as you rightly said, in BRC the opportunities are given on a plate. You look for any specialty, you will have somebody in that in our institute. But outside in the universities, also they collaborate very widely. If you look at some of the papers, the seminal papers, you will find 35, 40 authors writing a paper, because they collaborate within the like when you mentioned about University of Missouri, Dr. Ramurthy was also there. Many of my colleagues they have a very strong bond of working between the biologists, the clinical people, and the chemists. So they know that unless each one supports the other, you will not go further. So that idea is already there in the beginning, and they collaborate right from. They know if we have to develop this, you will have to NMR person will have to do all these things. So the person from the organic chemistry lab, metallo-organic chemistry lab becomes one part. Then the person who does crystallography is also there a part. Animal house managing person is there, and then the clinician is there. So they always, which is a radiopharmaceutical development and research, they always have a big team. They know where to connect and how to work in collaboration. Not just in the same university campus, sometimes it is in the same state, sometimes within the country, sometimes even between the countries. So it has to be a collaborative work when it comes to the development of radiopharmaceuticals. Almost everywhere they really recognize it. They recognize it and they work together. And the egos are set aside, and they have to work together so that they can progress. They can publish first, they can progress, they show the results, and they move on.
Okay. There are some questions coming up on both YouTube and WebEx, and I'm sure that you will be able to take few of them. One of them is in the current scenario where we are knowing about the pandemic and about the vaccines being developed, and a lot of debate about trials going on in the internet and TV and so on. So one question being asked is, do the radiopharmaceuticals undergo similar clinical trial procedures as done with normal drugs?
Yes, definitely they also undergo clinical trials, but it is a special formulation. So the one major difference between radiopharmaceutical and regular pharmaceutical is these are administered in very small quantities, number one. And they are all, one time, mostly the diagnostic. Suppose if it is a BP medicine, it's a very different kind of drug compared to radiopharmaceutical, which is injected once to get a picture of the heart or a picture of brain, etc. Or even if it is therapy, maybe 2 times, 3 times, 4 times. It's not like every day somebody is taking the drug. So the idea, the amounts used are very different. That is number one. Number two is it is short-lived and it's the one has to look at whether it is efficacious or not. But in spite of that, the clinical trials are very extensive for radiopharmaceuticals also. They go in stages, stage 1, stage 2, stage 3. The numbers, the variations, all the statistical parameters, they're all taken care, and which is why it is very expensive to bring out a radiopharmaceutical. One may work in the laboratory and show some molecule is excellent, but to bring it out with the FDA approval or drug controller approval, it's a different issue, because you will have to have lot of patients saying, registered, done a thorough clinical trial and show the results, which makes it all very expensive, which is why maybe there are some molecules which are fantastic, which have not become common radiopharmaceuticals due to this reason. But the rules are there, clinical trials have to be done. There are clear rules for each kind of radiopharmaceutical, what all need to be done, how many patients, what has to be shown. So European Nuclear Medicine Association and the US Nuclear Medicine Association have very detailed descriptions on these. They can be found in their websites also, and they do work in that fashion. Yeah.
Thank you. We'll wait for some more questions to come up from the audience. So meanwhile, I have another question. So bottom line for a common person is cost. Now we see the same discussion with regard to the vaccines also today, that whether it is affordable to a common person, apart from being efficacious. Of course, once one understands the test to be efficacious. Now, there can be it can be research direction, is it not? How to reduce the cost. But now if you're looking at very tricky kind of systems where you require expensive accelerators or a nuclear reactor to be available to you for producing something, how does one finally look at bringing down cost?
Yeah, that is, of course, it is a challenge, I agree. So again here, collaborations. Because if somebody is having the cyclotron already, then it is a good thing to collaborate and seek for some time in the using the machine and producing that. Otherwise, if we are thinking of some exotic isotopes which require, let us say, 100 MeV proton to be hitting the target, then the cost does become a big issue. Where in many countries, the national facilities, they have these big instruments installed, and so the nation sort of helps in reducing the infrastructure cost, and it's only the target and other things. But if a researcher in a small institute or in a university wants to work on that, it is going to be a big challenge. Maybe it is possible through collaborations, one, or one will have to approach the bigger, the resource things like maybe BRNS, things like that, where one can look for creating some facility where the work can be done. But if it's a huge cyclotron, it is going to be a challenge. Now the cyclotron in Calcutta has been established and it's functional. So people will have to look whether existing one within the DAE family or within the Indian government can be approached and used for specific research.
Thank you. So there's one question about waste management. Somebody wants to know whether there can be any unique or tricky questions of waste management in development and use of pharmaceuticals, radiopharmaceuticals.
Radiopharmaceuticals itself uses isotopes which are necessarily short-lived when it comes to diagnosis. When you saw technetium, fluorine, they're all 6 hours, means you can keep the whole thing for some time and it can go. But when it comes to the production of isotopes, particularly if the isotopes are produced through fission route, like for example, people who are familiar with the nuclear fission, you know that if uranium is undergoing fission, you will have 2 humps, one at close to 100 and close to 130. And if you are only looking at one isotope, let us say molybdenum or iodine, and then the rest of it, which is all waste, it will be a lot of waste with a lot of radioactivity, some long-lived, very long-lived, and so those can be it's not a problem, but it has to be addressed. So it is isotope production or any facility will have to address the whole thing in a holistic manner. It's not just I produce, I use whatever I want and rest of it can be left there. No, one will have to address the all the other isotopes which are coming out of it, how it is going to be stored, how it is going to be disposed, etc. So that waste management angle will come when it is fission produced isotopes or when isotope production is done, whether long-lived isotopes are also produced which are not desired, they will have to be managed. They will have to be separated, stored, and disposed of in the right way. So when it comes to isotope production, the people from the regulatory authority also become a part in approvals and in the processes.
Okay. Thank you. So just another question which comes to my mind, that in the case of vaccines, for example, there are many who point out that India is among the largest producers in the world, and we are having the pride of supplying vaccines across the world. How is the situation with regard to special radiopharmaceuticals? So is there any new developments which have taken place in India, any new compounds which have been developed, which have become a benchmark, or are we having any advantages in any of these domains?
New in the sense, when it comes to entirely new molecule, I wouldn't say that we have, sort of made some big breakthrough or anything, but we are at par with many countries when it comes to working with labeling of antibodies or peptides, etc. We have caught up with what is happening, and right from the production of isotopes to making it and supplying it. For example, lutetium, when I mentioned, it is an excellent isotope for therapy, but it took, I think very recently, lutetium thing was approved by FDA in US, and it was being used in pockets wherever it is being produced and used in Europe. And so India can claim to be one of the countries which is among in the top few who is able to indigenously produce and make radiopharmaceutical, supply and use. So this kind of production as well as processing, radio labeling, making it even into a kit if necessary, and supplying to the hospitals, this has been possible because of the nice structure we have within the BRC, the reactor, the BRIT, the people, the regulatory authorities, everything, and radiopharmaceutical committee, which looks at what is being prepared and giving the very good suggestions to improve, looking at the quality and then approving. So we have some uniqueness in the sense not every country can claim to be quickly adopting and supplying. But at the same time, when you say we have developed something which is groundbreaking, I will say no, not yet. I hope it can be done. We don't have this, when it comes to monoclonal antibodies, many times we have to buy, and not everything can be made from scratch. So peptides we buy, but then we do label them and supply them. There are a lot, when we look at the product list of BRIT and what is being supplied, even from what is in the advanced stage of development in BRC, they're very impressive compared to many other countries. So to that extent, I would neither claim that we are at the top and we have some many things, but we have several things which have been recently introduced, and we have trying to adopt and try to give those to the patients. For example, for the thyroid, not just thyroid, iodine labeled MIBG for therapy, it is not always available everywhere, but we are being able to produce and supply in a very good manner. I think I should congratulate all the young scientists and BRIT and people who have helped in automation, people who have helped in clearing it, etc. So I think it is a good situation. It has potential to improve also, but yeah, that's it. If we can separate alpha actinium-225 or whatever in a bigger scale, if the DAE community can come together and see whether we can see, right now there are only 2 or 3 sources of actinium, in Europe, in Russia, in the US. So if we can come up with something like that, that will be something fantastic, because the results are very good. People are looking at our actinium source, but it's a different. I cannot say whether it is feasible, whether it will be done, but opportunities are there, and people who are much more who have oversee the whole program will have to think of it. So those are the opportunities which can be explored.
Okay, one last question from me. We'll see whether there are questions from audience more. Do you come across patent or IPR related issues here also, or is it that people are very open-minded? As soon as they make a radiopharmaceutical, they announce to everybody how to make it and all the tricks so that others can benefit?
Think till now we have been like that. We have been open, and we look at publication, and we have been doing it. We have not. I don't think we have kept any trade secret kind of thing, and it has been our mindset that it has been our mindset from the beginning that atomic energy, nothing is patented and things like that. I don't think we have such a.
How about other countries? It's the same practice.
Not always. Sometimes they do have. See, they give, particularly if it's a new molecule, they have some part of it will not be mentioned. They may publish only what is the end product, but they won't say how it is made. It used to be like that. Yeah. Even now, I think some of the molecules are kept as a patented thing. But now, new molecules coming out as big drugs is much less now, because, as I said, clinical trials are extremely expensive. So that is another reason why you don't find so many people work with radio-labeled antibodies. There are, as I showed you, some 50 antibodies. A lot of patients are getting a radio-labeled antibodies for treatment, but not as a regular clinical. They are all what is known as orphan drugs, or investigational drugs. So that is one way out to treat people with radio-labeled molecules without making it into a huge drug with a brand name and all that.
Fine. I think at the moment I do not find questions from the audience. So I would like to thank you, Dr. Meera, for sparing time to prepare an excellent presentation. I know I have been putting pressure on you for the last a few months, and besides your all other activities, you have spared time for us today, and it has been a very excellent presentation. And the idea was to give a message to the students and faculty of HBNI that this is really a subject where various disciplines have to play a role together, and we definitely require collaborations between all the institutes with different specializations to be able to realize the product. And your presentation brought it out very explicitly. Professor Grover wants to ask a question, perhaps.
No, thank you. It was very good lecture, and thanks to you for arranging it, and thanks to Meera for delivering the excellent lecture.
Thank you so much for your kind words, but I know that I rushed through several things. I cannot explain everything, and I'm not sure. I'm sorry if for some people things were a bit rushed and not very fully explained, but I'm happy to communicate.
As I said, Meera, this is an appetizer, and we will probably ask you to give a course on radiopharmaceuticals for where you can take more time to explain everything. Thank you very much for joining us today. And just as I announced in the beginning, our next webinar will be on 20th by Dr. Arun Srivastava, and I wish all of you find time to join. Thank you very much.
Thank you.